I'm John Gribben. We're at IWNHL in beautiful sunny San Diego. We've had 2 days of really interesting sessions, and we've just wrapped up a session looking at novel agents in the lymphomas. Actually, I just had a comment from somebody a minute ago talking about how they hadn't been at this meeting for a few years and that it used to be much more scientific and they thought it was more clinical. And my view was that, in fact, we've now got drugs that hit the targets that we were working on several years ago. So it kind of does feel that that's where we're going with the targeted therapies you were talking about here today. Mm-hmm. Now, we did start by talking a little bit about somewhat less targeted therapy, that is the IMiDs, so looking at modulating the immunological effects. And of course, a drug which clearly has efficacy, seems to have increased efficacy with antibodies, but also, of course, unmasked some other toxicities with some of the other agents that you were talking about also. So where do you feel that the kind of immunological approach alone compared to the more targeted approaches are going in this kind of field?
I think they're probably heading towards combinations. But one of the things we heard today from Dr. Fowler was that we need to be very cautious about mixing these drugs together. And sometimes your intuition to add drug A and B seems obvious, so go ahead and do it. But Nathan shared with us some data regarding the combination data of lenalidomide with idelalisib, where they had substantial unanticipated toxicities that led to early closure of the study. So I think trying to find strategies that are less chemotherapy predicated. We also heard about some interesting data where they shared the R-squared data in mantle cell lymphoma up front, and it had an astoundingly high overall response rate compared to what we're used to seeing in the relapsed refractory state. So maybe there's something different that's going to portend better responses by using these things earlier instead of chemotherapy. But I think the one glaring message for me is that these things need to be combined carefully in the context of a clinical trial and not just haphazardly combined because they satisfy your intuition.
Well, I guess you could say it might be the same kind of thing that we see with the PI3 kinase inhibitors when we use them up front. That is, a more robust immune system that hasn't already been potentially damaged by previous chemotherapy and by longer exposure to the disease may actually have either a better effect if you're using an immune stimulatory drug, but may also unmask the toxicity of autoimmune type phenomena associated with the use of these agents. Mm-hmm. So the PI3 kinase inhibitors are one whole group, but obviously the BTK inhibitors have completely revolutionized diseases that you and I kind of think about a lot, Simon, and you kind of gave us an overview of where we are. Now, clearly, Ibrutinib, the leader in the field, the drug which is already licensed and approved. You were presenting data on kind of now very long-term follow-up in that kind of setting and no real picture of increased toxicity with time.
I guess that's where that class of drug is, well, it's not unique, but that's where it doesn't have the same problems that the PI3 kinase inhibitors do. That long-term side effects are not emerging. You keep the patients on the drug, and actually incidence of infection seems to be going down, with the possible rare exception of when we're using it in CNS lymphoma. Immunoglobulin levels don't fall. You're not seeing emergence of new side effects. In fact, it's not been shared yet, but if you look at the side effects people worry about, the bleeding and the AFib, if you don't get that in the first 4 or 6 months, you're not going to get it. So there's a group of patients that get it and some that definitely don't. And with further longer term exposure, they don't get it. So it's an early thing, and you're not getting an acquisition of longer term toxicity.
Yeah, it's like a different pattern of people coming off drugs with Ibrutinib compared to idelalisib. It's that you've got those people that don't tolerate it established quite quickly with Ibrutinib, and then they tolerate it very well, whereas you get the accumulation of more toxicity over time that appears to occur with...
Yeah, and I'm somewhat perplexed by this high percentage of patients who are coming off Ibrutinib from intolerance, and I think that reflects a CLL population, untreated, asymptomatic, getting a drug and getting some toxicity and therefore stopping. Where if you're using it post-chemotherapy, which is the place I'm using it, nobody ever comes off the toxicity because relatively this drug has got no side effects as far as the patient's concerned. So it's dependent on the disease you've got and the relative state you're in when you start taking these things.
And the potential other alternatives that you have, because of course in
Exactly.
the CLL field
You've got other
already
options,
we've
of
got
course.
other options which outside of a clinical trial you don't yet have in the mantle cell lymphoma setting. And then wasn't focused on particularly here, but there was a lot of over the session about the whole use of these agents in the more aggressive lymphomas where there's B-cell receptor signaling as part of the pathway. Do you really believe that, you know, for ABC type lymphomas that agents like Ibrutinib or other BTK inhibitors are really going to be absolutely part of what we're doing going forward?
Well, it's interesting. When we did the early trials with these drugs, so BTK inhibitor, when you treat a diffuse large B cell, you see a response. You see a response. It's often very early, but it's gone at 6 weeks. So there's clearly some activity, but that's not enough. So that by itself gives a rationale to combine it with something else. You know, these drugs, they're great. In the context of CLL, you probably don't need to add anything. Waldenström's, you probably need to add maybe an antibody. Mantle cell, you need to maybe add chemotherapy. Diffuse large B-cell, you definitely need to add probably chemotherapy now. Maybe combination of novel agents is what's going to be there in the future, but you clearly need more in the more aggressive diseases. But there's certainly you know, you can see when you treat these patients, you see a response, but it's gone very, very quickly. So, and it's the same with primary CNS lymphoma. So you definitely need something else there. You know, there's cleverer people than me will tell you why these drugs work in certain diseases, not others. But so the rationale in the ABC subtype is clear. And you see that with the BTK inhibitors. Yeah.
Now the other issue, of course, that you focused on was the fact that pretty much all of the newer compounds all have the same binding site, and therefore it's not going to be like a Gleevec resistance mechanism of alternative drugs. So is there really a need for these other agents?
That's a good question. Well, from my perspective, you need more than one BTK inhibitor. I think there are 12 ACE inhibitors, maybe more. You know, you need some selection, and there are some subtle differences in the side effect profiles. If it's true that there's no cardiac toxicity with some of the second generation drugs, then you would preferentially use a drug like that with a past history of cardiac disease, because those are the ones that preferentially get the atrial fibrillation. So you can see a role there. But I think the main issue for me is it will drive down price. If you've got more than one drug, you will inevitably get competition. And that's the important thing. I mean, we live in a country where NICE has just said no to these drugs, and that's primarily because of cost. So if we've got more than one, then the cost has to come down.
Now, it doesn't seem that long ago we were looking at PI3 kinase inhibitors and thinking this was the best thing we'd ever seen, and until along came the BTK inhibitors. But kind of maybe light at the end of the tunnel with some of the things you were talking about today. So I think the major problem with the PI3 kinase inhibitors has not been the lack of efficacy, but the potential side effect profile. And you had a very, very intriguing story regarding slight difference that you see and a good explanation for what we've observed clinically. That is this very different toxicity profile with TG1202 compared to the other members of the PI3 kinase class.
Yeah, it's interesting because a lot of those toxicities, as you know well, emerged kind of late, and especially the colitis-related issues. And so as the TG program emerged, and we did not see that same level of toxicity in terms of LFT increases, pneumonitis, or colitis, the default opinion was, oh, it has to be less potent. Oh, it has to be a PK thing. And when you delve down, at least from my humble perspective, and you look at the concentrations in plasma and area under the curve and the low nanomolar efficiency of these drugs, they're really, when you look at the case of I against the enzyme, they're low nanomolar drugs. And we're achieving, you know, tens of micromolar of these drugs in the blood. So the spectrum is huge. And so early on I had a suspicion that it wasn't going to be as simple as just change the dosing or the interval. And so Dr. Deng in my group and his graduate student Mark Lipstein really, you know, had the insight to actually be very translational in their approach to this. They looked at the data that was emerging on the trial we had, seeing activity in large cell lymphoma, which was unprecedented for the class of drugs, and then trying to understand that in the context of their focus, looking at the importance of inhibiting translation for c-Myc. And in that process, they uncovered that TGR, in fact, hits a dual kinase. It inhibits the delta as well as any of the other inhibitors, but it also appears to inhibit this CK1, which turns out to be quite fortuitous because it happens to be a major regulator of protein translation through the EIF4F complex. So now the next step experimentally will be trying to connect the dots. So we have a potential explanation for differences with regard to activity in large cell, but how do we explain the impact of CK1 on the lack of immunologic side effects? And so that's going to take a lot of creative thought, but there's already a substantial literature that gives us clues that around which we can generate testable hypotheses. And I'm pretty confident that some of those immunologic effects that we don't see with TGR are not a consequence of failure to hit the target, but are rather effects counterbalanced by this unique dual kinase inhibition.
You've got studies open in diffuse large B-cell lymphoma using this compound. Are you having the opportunity in those studies to rebiopsy and look at things like MYC protein levels and determine whether it's really...
Yeah.
Okay, so...
Spectacular point. That's one of the trials we're doing now is going to be to look carefully at the kinetics where we can in terms of getting tissue from a patient. And we have big mouse experiments looking at that because, one of the huge unmet needs in aggressive lymphoma is double hit.
Sure.
And
we've always been told this was an undruggable target.
That's
and
right.
So hitting it indirectly
Right.
may be the way to do that.
This may be less sexy, but it may be more effective. And so if you can use strategies like that turn it off quick, because MYC contributes not just to a proliferative rate, but it contributes to chemoresistance. And so if you can downregulate MYC at just the right time with the chemo coming in, you could have an impact on the disease.
Can I just ask, so what about duration of therapy? I mean, the PI3 kinase inhibitors clearly are active, and I think they've been slightly snookered by the fact you can give Ibrutinib forever. Yeah. So they've tried to do exactly the same. I mean, surely they should be re-evaluated using shorter courses. Is that the plan with the TG drug?
At the moment, in the DLBCL, the Unity DLBCL study, and John's working on one in CLL, the plan is to give the PI3 kinase inhibitor plus ublituximab for I think up to 2 years or something. So there's a finite, but it is a protracted dosing. But it's not a pulse like 6 cycles and you're out. It's more protracted because it's better tolerated and you don't know. But ultimately, we need to understand those influences to be able to learn how to use the drug without all the economic toxicity.
Now, you also showed that very intriguing data of synergistic activity with the TG1202 and Carfilzomib. Any plans to explore that clinically?
That's happening now too.
That's happening now.
Yeah, so we just opened that study now. We should be putting our first patients on.
Which disease setting do you think that would be potentially most effective?
So again, that's being done. The phase 1 is going to be all comers just to get to MTD. But once we're at MTD, the phase 2 is going to be looking at the impact of TGR-1202 plus Carfilzomib in patients with aggressive, multiply relapsed diffuse large B-cell lymphoma, where we're going to try to get patients, just a small number to get a hint, but just to try to do some of the correlative studies you alluded to. So we can try to look at the kinetics of MYC change as a function of treatment to figure out how we're going to build on it. I don't think that TGR-1202 plus Carfilzomib is the next, saving gift in large cell lymphoma. I think it's going to give us insight into how we can optimize those drugs and maybe an upfront strategy with chemotherapy to see if we can't downregulate MYC prior to the induction.
Sure, that'd be intriguing to be thinking about. We could have BTK inhibition for the ABC subtypes and a novel PI3 kinase Carfilzomib combination for the poor risk GCB subtype. So using different attacks on the same sort of pathway, we're thinking about hitting very different vulnerable spots in the drugs in that setting.
Yeah.
So there you have it. I think real translation through from very basic observations made a number of years ago on the importance of these pathways to identify druggable targets, to then identify also ways in which these drugs are often working not exactly on the targets on which we thought. So I think we're all excited to see where this field takes us and where those studies that are ongoing will go in terms of improving our outcomes for our patients with B-cell malignancies.